Today we are going to see about the post-transcriptional modifications. First, introduction. It is a primary transcript made by RNA polymerase. It's normally undergo further alterations called post-transcriptional processing or modifications. And primary transcript is nothing but is a RNA synthesized from the DNA. After this synthesis, mRNA undergoes some processing in eukaryote that is known as post-transcriptional modifications. In prokaryotes, mRNA transcript directly from DNA template and used immediately in protein synthesis. In eukaryotes, primary transcript must be processed to produce the mRNA. It occurs in nucleus of the cell. And the next, these post-transcriptional modifications of RNA accomplish three things. First, it helps the RNA molecule to be recognized by molecules to mediate RNA translation into proteins. During these post-transcriptional modifications, the portions of the RNA chain that are not supposed to be translated into proteins, and these are cut out of the sequence. And the next, without post-transcriptional processing, the protein synthesis could be significantly slowed since it would take longer time for translation machinery to recognize, and significantly more RNA would have to be unnecessarily translated. And these are some terms used in post-transcriptional modification. 3' splice site. It contains an adenine next to the guanine base at the 3' end of an intron, and 5' capping. It is attachment of GTP molecule to the 5' end of the mRNA. And then 5' splice site. It contains an guanine next to the uracil base at the 5' end of an exon. It is coding regions, and the sequence that are preserved in primary RNA transcript splicing that will be translated into proteins in the cell cytoplasm. Introns, it is a non-coding regions, and in this primary RNA transcript that are removed and degraded during primary RNA transcript splicing. Guanyl transferase. It is the addition of the GTP molecule to the 5' end of a primary RNA transcript. Polyatyl. It is the addition catalyzed by the enzyme poly-A polymerase that recognizes the sequence AAU AAA. RNA splicing. In this, introns are removed from a primary RNA transcript and exons are joined together to form a mature mRNA. And these are the three methods includes in post-transcriptional modifications. 5' capping, additions of polyatyl splicing. First, we move on to the 5' end capping, and it is the attachment of GTP molecule to the 5' end of the mRNA. And in this, the capping process replaces the triphosphate group with another structure called the cap. The cap is adding by the enzyme guanyl transferase. This enzyme catalyzes the reactions between the 5' end of the RNA transcript and a guanine triphosphate molecule. And this, what is the use of 5' capping means? It prevents the mRNA from the attack of 5' exonuclease activity and gives stability to mRNA, and it also helps in translocations of mRNA from nucleus to cytoplasm, and it also helps in binding of mRNA to the ribosomes. And then the capping protects the 5' from enzymatic degradation in the nucleus and assists in export to the cytosol. In eukaryotes, mRNAs lacking the cap are not efficiently translated. And then polyatyl, and then it is in this, it with the help of enzyme polyadenylate enzyme, there is a intrinsic signal AAUAA. These are the termination signals. After this signals, it releases RNA, and there is attachment of multiple AAA or adenine molecule near about 80 to 250 nucleotides, and these are added with the help of polyadenylate enzymes. And this polyatyl appears to protect the 3' end of the mRNA from 3' 5' exonuclease activity. And then histone interferons mRNA lack polyatyl. After the mRNA enters the cytosol, the polyatyl is gradually shortened. And the next, the green color indicates the 5' capping, and this red color indicates the 3' polyatyl, and the center is mRNA. And then next, splicing. Splicing is the removal of introns. There are two main mechanisms in splicing of mRNA primary transcript. They are exons and introns. So the coding regions is exons. Coding region of mRNA is exon, which is responsible for synthesis of functional polypeptide, and the non-coding region is introns. And the molecular mission that accomplishes the task of splicing is known as the spliceosome. Small nuclear RNA molecules that recognize splice site in the pre-mRNA sequence. In this, the excised intron is released as a laterite structure which is degraded. And in this diagram, this is exons and introns, and the center portion is an intron, 5' region and 3' region, and this is intron. This is 5' splice site. This is 3' splice site. This is known, and then this A is known as branch site. After recognizing that is attack on 5' site, and there is a cut in 5' splice site. After this cut, this lateral structure is formed, and then this exons attack 3' splice site and will remove the laterite. Now the spliced mRNA is formed. And then next, alternate splicing. It is adapted for the synthesis of tissue specific proteins, and this produce multiple variations of mRNA, and thus diverse set of proteins can be synthesized from a given set of genes. And then from this, introns are removed from the primary transcript in the nucleus, and then exons are ligated to form the mRNA molecule. After the removal of all introns, the mature mRNA molecule leave the nucleus by passing into the cytosol through pores into the nuclear membrane. These are the reference. Thank you.